Today I want to talk about increased crop yields through improved photosynthesis. I'm sorry about my voice, I lost it last night. I'm doing my best. I run through these slides quickly, the first three because you know them from yesterday. Yield increase is important because of the growing world population. Second generation traits are a big opportunity, and what's done now by the ag industry is high throughput screens, large investments in this, and what the company Benson Hill Biosystems is doing, where I'm working at, is focus on photosynthesis to increase crop productivity. And so we asked ourselves that question, and interestingly 2006 also Steve Long and Don Ord and the people at UIUC as in Illinois asked the same question: can improvements in photosynthesis increase crop yields? It's a nice paper on this, and essentially Steve Long is the key author, say yes, it does. It has a large potential, and they broke it down a little bit what the different steps are: improving Rubisco, increasing this Kelvin cycle, and so on. And in the end, the yield potential of a plant, which is so to say the yield a plant gives under optimal growth conditions, if there are no diseases, no stresses, and so on, is largely defined by parameters, which are the first two here. There's epsilon I and epsilon, what is it? Oh God, my Greek is poor. These two here are actually quite optimized through breeding, while the conversion efficiency, the conversion of light energy into, in the end, carbon, is far away from the optimum and can be further improved. And so this whole equation is actually used by Steve Long, but was developed earlier, quite well accepted in the photosynthesis world. So the answer is probably yes, and probably significantly you can improve crop yield if you improve photosynthesis. And I have two papers here which are not from us, but have been published recently, which visually confirm that it works. Here, this one is from the group of Christine Renz in the UK, who just 2 months ago published a paper on overexpressing several enzymes of the Kelvin-Benson cycle, and also one of the genes is actually a carbon transporter in tobacco, and they show a very nice and significant biomass increase. Obviously, tobacco, it's about biomass or leaf biomass. And another paper, this study goes back certainly more than 5 years. Andy Pereira, at that time in Virginia Tech, and then later on at his new university, where is it, in Arkansas, I think, worked on that HYR genes, also HYR, which is a transcription factor which seems to upregulate several parameters of photosynthesis, and he showed very nicely that this overexpression, a constitutive overexpression, leads to increased drought tolerance. These are the data which I'm showing here, and in that paper, and that Nature Communications paper, he also shows other aspects. So quite nice. From an IP, as an intellectual property perspective, these are not very well protected, but as a result, they work well. Our company focuses on this part, also on Kelvin-Benson cycle, on all other aspects of photosynthesis. That's where we try to identify limiting factors and increase crop productivity. And this requires often multi-gene constructs, so it's a little complicated. It's not as straightforward as doing Roundup Ready or BT. And on one slide, the company summary, also we branded ourselves the photosynthesis company, so that people understand what we do. And I think I went through most of it, and you can imagine that if you understand photosynthesis, it's actually quite broadly applicable across different crops. So in the end, there is of course C4 and C3 photosynthesis as crop plants, right? Like corn and let's say sorghum, sugarcane as C4, while rice, wheat, cotton, etc. are C3. If you have time at the end, I can also say things about non-GM. What we do can also be in the end a non-GM product, not just GM. Relatively young company, 2.5 years old, and we have already 3 commercial partnerships in place. with these symplots, CTC. A symplot is in potato, CTC in sugarcane in Brazil, and Limagrin is obviously globally in wheat and corn. Okay, so what I think that the key success factor, and I think we are doing quite well, is really an integrated discovery platform where we start, so to say, at 1 o'clock with a deep knowledge about photosynthesis, and then the next 3 bubbles here are important. We study the gene regulatory networks, also through large RNA-seq experiments, also transcriptomics experiments in the plants under different growth conditions and different developmental stages to identify networks, regulatory networks. And also, networks are not stable. If you change the environment or if you look at different developmental stages, they are different. And these networks change, and it's very important to understand how they change, which ones are robust and which are not. So to identify the regulators, to identify under which conditions you have to express them in the crop plant. Okay, we combine all these data. We have smart people who do computational biology, and in the end spit out some candidate genes. Okay, express this gene with this promoter and this specificity. We test it in model plants, so we have C3 model plants like rice and Brachypodium. We have C4 model plants like Ceteria, and have phenotyping in place, and whatever works in the model plants we put into crops, also into corn, well, rice is already a crop itself. We also work in soybean and wheat and with partners in other crops. Okay, and obviously test them in the field as soon as we can. The key for this here is this company is actually a spin-off company of the Donald Danforth Plant Science Center. I'm not sure if you know it. It's really a very important plant science institute in the US. And Tom Brudnell and Todd Makler, who are PIs at the Danforth Center, are CSO and CTO of our company at the same time, kind of have a double life. And that gives us access and saves us money and gives us access to infrastructure and also to inventions from the Danforth Center. I skipped that slide because I told you we are testing things in model plants and go into crops in order to speed up the process. So what did we really do? Here's the word PSK-based. It means just photosynthesis knowledge base. Is a fancy term which we identified to brand all that the databases which we produce and the algorithms used make sense out of it. So I give you 2 examples of what kind of gene regulatory network, or actually RNA-seq experiments we performed, which are sources of our gene discovery. Here we looked at C4 plants. Also on the top you see sorghum, corn, and then Ceteria. Ceteria is a model plant, but also C4. We looked at different sections of the leaf. A C4 leaf is very simple. The base is old. Sorry, the base is young. The tip is old. No? You don't really have to go for young leaves and old leaves. One single leaf has old and young tissues at the same. And we isolated, in this case, mesophyll and bundle C cells. And from these different cell types, we performed RNA-seq experiments so that we have different developmental stages and different cell types. Another experiment which we did was in wheat, where we grew wheat plants under different CO2 concentrations under very controlled conditions, like we heard it yesterday. Again, here it shows 4 different developmental stages and 3 different CO2 concentrations in order to then identify, or to look at all the transcriptome, and wheat quite big, you know, more than 100,000 genes are expressed, focused on the photosynthetic subnetworks. You know, there are known photosynthesis genes, and those which are co-regulated with them are likely to be somehow involved in photosynthesis. And this picture is actually quite interesting. If you look at how these genes which are associated to photosynthesis are working under low CO2 concentrations, and how these networks cluster under high CO2 concentrations. That is totally different. It's totally different. The increase of CO2 really makes the gene regulatory networks absolutely different. And that's key, because if you want to find the regulators, they are different under high CO2 compared to low CO2. I think this is also a good fundamental research result. which should make people think about the experiments. I skip this slide and go show you some examples. I showed you that slide about the C4 leaves, yeah? So we identified transcription factors which seem to be regulating photosynthetic networks. And so what we did, we identified 8 of these transcription factors. We expressed them under the control of 4 different promoters. What I called here A is a constitutive promoter. The other ones are leaf-specific with different specificities. And then grew these, transformed these into our model plant in a C3 model plant, Brachypodium, and had a whole series of interesting phenotypes. On the top you can see one which has a delayed flowering but really grows much faster, much more biomass production, while the one on the bottom have in fact both. So on the left is always sorry, I forgot the labels. On the left is always wild type, and here is the transgenics. Here also wild type transgenics. So they have both increased biomass and increased seed yield, and I forgot that slide. But this also goes along with increased photosynthesis measurements in these plants. So there's a correlation between the growth and the photosynthesis measurements which we performed. Then we have more advanced lead genes, which when overexpressed show a nice effect. This one here is a CO2 transporter, which increases the CO2 concentration in plastids. It comes from cyanobacteria, actually. And when it was transformed in soybean, and there are also field trials already available in soybean, and these show not dramatic, but significant increases of 15 and 7%. 15 was in greenhouse and 7% increase in field trials, multiple years. That is quite promising. We have I skipped this one, it's not really photosynthesis. This gene here I find interesting because it is not directly photosynthesis, also not acting in the source tissue, but in the sink tissue, in the corn kernel actually. It's involved in starch biosynthesis, and what it does, it's a starch synthase, which was modified so that it's less heat labile, so it is more stable under heat conditions, and this seems to be the limiting factor of starch biosynthesis in the corn ear actually under high temperature stress. And indeed these plants show very much increased ear weight compared to control conditions. Last but not least, I have one example here where this is an approach. It's not published yet, so I can't really say exactly what it is. But it is very C4 specific. Also we enabled these C4 plants. In this case, we tested this in Ceteria. These are the results from Ceteria. When overexpressing this gene, they have several ways to actually transport the C3 and C4 metabolites from the bundle sheath from the mesophyll into the bundle sheath cell. Usually C4 plants have one subtype. These are kind of limited, and we enable them to have several routes to really move more carbon into the bundle sheath, and which in the end means the decarboxylation takes place and CO2 is released, and then you have higher CO2 concentrations around Rubisco. And the effects we saw in our model plant were striking, like 130% dry weight as a biomass increase, or 400% increase in seed yield under very low nitrogen conditions. This is quite interesting. Under higher nitrogen conditions, the effects are still there, but less striking. So that's one of the most promising candidates, certainly. I told you this is applicable to many crops, so we can skip this. And obviously we are small companies, so in the end we have to partner with the big ones who have seed market and who really do have breeding programs and so on. So I thank you very much.
Questions, comments?
Yeah, please.
I was kind of asking you yesterday, like, what is the acceptability of these kinds of technologies commercially?
Yeah, I mean, some crops, as in some crops, there's certainly no doubt, and in some countries, also like in corn and in soybean, also in cotton, these are GM crops anyway, so the acceptability is high. in wheat, interestingly, Limagrain signed a large collaboration agreement with us in wheat, and they are working on transgenic wheat. Well, knowingly that the development times are beyond 6, 7, 8 years, and hoping that the acceptance scenario changes then. I mean, what I didn't say is, particularly in crops like wheat or rice, also food crops really, where you eat the kernel itself, or more or less processed, unprocessed. We wrote a letter to the USDA, APHIS, and described experimental setup where we, almost all our candidate genes are actually from crops. They are not mysterious genes coming from bacteria, except for this one CO2 transporter, which I mentioned. So there are other ways through genome editing to, for example, to activate the already existing genes, just to overexpress or to increase the expression level of the already existing genes, which you could do through breeding, but often enough it doesn't work through breeding. Anyway, this approach, USDA responded, it would be considered non-GM if you do it right. You have to do biolistics and not agrobacterium, because agrobacterium-mediated transformation is a plant pathogen, and then it's already considered GM. But of course, that statement from the USDA is limited to the US. No? It doesn't help us in Europe. Oh, sorry, that's my answer. Yeah.
The last question?
Yes, please.
Yes.
I just want to know that if this the improvement in photosynthesis for the plant, is it applied on a commercial scale?
Yeah, it is. We are obviously a commercial company. Well, at first, when we are just 2 years old, 2.5, so obviously our results are, most of them are in model plants. I showed you a few crop data. So applied usually by our partners. They transform it into crops, or in corn, we do our own corn transformation, but we just performed the first field trials this year in corn. All the field trial data I showed you actually came from the institutes where we licensed the technologies in. Some genes are in-licensed, some come from our own discovery pipeline. Yes, it's commercial, absolutely. Not yet, but that's the target.
Okay,
Yeah.
and C4 plant, more promising than C3 plants in this
Well, what a C4 or C3, what is more promising? Well, obviously the potential to increase photosynthesis is actually higher in C4 and C3 plants, sorry, because C4 is, at least for the CO2 concentration, quite optimized, but there are still rooms to improve also C4 photosynthesis. I showed you that result in Ceteria at the end, which shows that also it's a C4 plant, should be doing quite well, but you can improve photosynthesis and growth. So yes, some things are working in both subtypes of crops, of plants. Some are C4 or C3 specific. It's too difficult to give a uniform answer. Yeah. Okay.
Thanks so much, Lily.
Thank you.